Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show to bring you another really fascinating guest for you today involved in science and technology and medicine, creating a better tomorrow for many people out there. Today on the show, we have the honor of being joined by Thijs Spoor, who is the Chief Executive Officer at Perspective Therapeutics. They are a clinical stage radiopharmaceutical company. They're pioneering these innovative treatments for cancer, and the company's proprietary technology utilizes this really interesting alpha emitting isotope known as lead-212, which we'll be talking about, to develop really powerful radiation directly to cancer cells via their high affinity targeting peptides. Prior to this, Thijs was a chairman at Verifi H2O. He also held the position of Chief Executive Officer at Viewpoint Molecular Targeting and KBP BioSciences. And prior to that, Thijs also served as CEO of AzurRx BioPharma, developing targeted therapies for GI diseases, as well as the CEO of FluoroPharma, which was focused on the commercialization of these really novel PET agents for cardiovascular, oncology, and neurology arenas. Thijs has also worked in strategy consulting at Oliver Wyman as a biotech analyst at the JP Morgan, also a medical device analyst at Credit Suisse. He began his career at GE Healthcare, serving as director of strategic marketing, and played key roles in that company, shaping their strategic direction. Thijs did his undergraduate degree in pharmacy from the University of Toronto, as well as an MBA in finance from Columbia, and we're honored to have him. A lot to get into today, really exciting topics. Thijs Spoor, welcome to our show.
Hey, great. No, thank you so much for having me. It's great to be here.
Yeah, I'm really looking forward to these topics. Before we touch on anything radiopharma or nuclear medicine related, Thijs, tell us just a little bit about your background. You and I are very similar. We both are pharmacists undergraduate. We both went to business school. We both hopped into the pharma industry. And I'd love to just hear a little bit about you and what got you excited about healthcare in general, and started you off.
Yeah, so my a lot of my family members are in the medical field, in social work and radiology and geriatrics. I went into pharmacy school and really wanted to see how to apply a great science training. And I got really excited about what was happening with nuclear. And so nuclear pharmacy, when I graduated in '94, there wasn't a lot of people going in that field. But I really liked the ability that if you can have the drugs go to certain tumors and not go to healthy tissue, you can have an extraordinary amount of either imaging potential or damaging therapeutic potential. And so that whole, direct application of technology is fantastic. I took a couple of detours to Wall Street, but having taken sort of companies public, it's one thing to understand the science and to develop innovative science. You always have to make sure the science works. Not all science works, and you're going to sort of go and try things out. I love the creative process. I love the fact that we can actually go and think of things that no one else has thought of and try them out and see if we can benefit mankind.
Awesome. Yeah, I do remember the one kid in pharmacy school that was interested in nuclear medicine as well, and we were all looking at him back like, what's that? But now clearly the space has come a really long way. Let's start out at the beginning here, because this is a topic that we've touched on a little bit on past shows, but really want to re-educate the audience. And I think a great place to start off would just be sort of on the core issue of the radiation particles, both the alpha and the beta particles that we've talked about in the past, and again, what they do do, what their benefits are, sort of the bottom line that we remember from previous shows, alpha particles, sort of equated to sort of very precise sled hammer, kind of devastating to the cells they hit, but they don't go very far. Beta particles, a little more diffuse, more reserved in terms of maybe larger tumors, and they spread out a little longer, can hit other cells and go a little further. Take us into the world of alpha particles and beta particles again, and sort of reintroduce us to what it's all about.
So, and there's the third one too, which is a gamma. And so in a lot of, and I bring that one up because when people think about radiation therapy, they usually think about gammas, and gammas will penetrate all the way through the body. And if this is, sort of a human body and the gamma goes through, and it's going to go all the way through, everything it's going to touch, it's going to cause some damage to. And the radiation oncologists are trying to figure out, can they actually get more energy cumulatively onto the tumor versus healthy tissue? But the danger with external beam, as they call it, is that it goes through all tissue, good and bad. And so you get an awful lot of extra dose to tissue that you're trying to save. By making a targeted radiopharmaceutical, it's almost like the switch from chemo to ADCs, right? You're actually going to, rather than making things systemic, you're going to bring that toxophore, in this case, bring that particle straight to the tumor itself. If you design it so it actually sticks to the tumor and nothing else, then you have the ability to wait and have the energy going straight onto the tumor tissue. Betas travel about 200 cell diameters, whereas alphas travel 2 cell diameters. And the difference is that betas will damage cells, but alphas will destroy them. And that's a really important sort of differentiation. And why we're seeing a lot longer duration of response and more impact from the alphas is that there's nothing that can withstand the physical hit of an alpha particle. It's a massive, massive particle. It blows the cell apart. It causes double-stranded breaks to the DNA, which the cells cannot repair. But for those that are more in the oncology field, there's another iteration with beta particles where they actually doesn't work in tissue that's hypoxic, right? So if you don't have oxygen in that tissue, then you're not going to get the free radical formation that betas are trying to induce. And so alphas don't have that limitation. Basically, if they exist, they smash apart everything they touch. It takes about 7,000 betas to kill a cancer cell. It takes 1 alpha. And so really, really big difference in potency. But potency is a double-edged sword, right? So if you're going to have those alphas hitting healthy tissue, they're going to cause a lot of damage to healthy tissue as well. And so unless you're really designing your molecules properly, you may not get the desired outcome.
And you mentioned something very important there, and just to reiterate, the alphas cause devastating damage, and the other important piece here, because even though we are in this era of sort of smart biologics and all this stuff, when we're dealing with radiation, we're dealing with physics. We're not talking about that protein or pathway or receptor which cells can evolve around ultimately. And we all know the story of sort of the classic Darwinian sort of drug resistance stuff. In the case of nuclear medicine and especially alpha particles, so this is something there's no resistance to, and I think that's one of the sort of important pieces of the radiobiology story that sort of I just want to stress again for our audience, because I think it's an important piece when people think about radiation that sort of overlooked, let's say, is part of the benefit here.
And that's a great point to bring up, because there are, the cancer right now has 3 different core pillars for how we actually try to help patients. Either you use chemo, radiation, or use the immune system. And so there's been a lot of energy getting the immune system work better. The nice thing about alpha particles that you don't see with beta or gamma is that alphas are really neoantigenic. So that alpha smashing into a cancer cell and then causing this burst of fragments causes a neoantigenic storm that gives the immune system something to grab onto. And so for any kind of treatment, some patients respond to only one of those 3. But when tumors get aggressive enough, you need to start adding things together. The one thing you're trying to watch is make sure you don't have stacking toxicities. And because the mechanisms of actions are so different, we actually don't tend to see stacking toxicity.
Absolutely. Let's talk a little bit about lead and lead 212, because as mentioned before the show, we have done episodes. We talked a bit about lutetium and actinium. Here we're talking about an isotope of lead that, and again, I tried to do a little research before, and correct me if I say anything stupid here. This lead isotope 212 is not itself an alpha emitter per se, but it becomes other things. I think that bismuth 212 and something else known as thallium. Take us a little through what this isotope is all about, because it has some unique properties to it, let's say.
So the really interesting thing about isotopes is that they go through natural decay, which is predictable. And so if the nucleus is unstable, it will then collapse into something else.
Yeah.
And on the way, it will give out usually a little bit of gamma energy, and in some cases a particle. And that particle can either be a beta or an alpha. The direct decay of lead-212 to bismuth-212 gives off a beta particle. So that's going to cause a little bit of damage. All it should not to go too deep in the physics of it, but every particle that we're describing here, or array, they can each have their own distinct energy levels too. And so there's a low energy beta that comes off from lead. And then the bismuth-212 will then fire off an alpha particle. And after that, then it decays and it turns into sort of non-radioactive lead. There's some other isotope cascade chains out there that lead to lots of toxic daughters that kind of ripple through as well. And so we like things that are pure decays because we know where it goes. And the great analogy is that it's like a toxic metabolite. In some cases, a drug will be active and then its metabolites aren't. In other cases, the metabolites can be active. And so in the radiopharmaceutical world, you care about where the daughter isotopes, what they are and where they go. Because ultimately, it's once it's gone down to a non-radioactive version, that's when all these decays happen. At a chemical level, the FDA calls what we do microdosing, and so they basically have acknowledged that the amount of physical material that we're giving is almost undetectable. But it's the radiation that's going to cause both the damage to the tumor and the potential damage to healthy tissue. And so we want to control not just where the first emission happens, but all possible decays and all possible emissions. So we've actually designed our medicine so that not just the lead, but also the bismuth daughter goes where it's supposed to go. And so if you can do that, if you can control where the daughters go to, you can then really increase your energy onto tumor and not have more energy off tumor.
Mm-hmm. And you have on the website a really cool chart, and again, comparing lutetium, actinium, and now what you're dealing with lead-212, and again, great potency, safety. You don't have the issue with the daughter isotopes, short half-life, and then it also talks about supply and convenience. Can you say a couple words, because again, we think about these isotopes and where they come from, and do we have to find them in a nuclear reactor somewhere, or what's the story in terms of just the amount of lead-212 that is accessible to you to do not just the therapeutics, but you also this theranostic stuff that you're involved in as well, which is we'll get to later on.
Yeah, so what I first learned when I went into the industry, I remember this, one of my mentors has said, so just remember, Chase, the easiest, cheapest, simplest way to make any isotope is do nothing, natural decay, right? So it's like, if you had an ice cube, can you make water? Absolutely. Just sit there, do nothing, and it'll turn into the next thing that goes down. And that's what we're really trying to take advantage of. There's a parent isotope called thorium-228, which is in relative abundance globally. And that thorium-228 will then go through a series of cascades down and turn itself into radium-224. The radium is what we actually ship around the world. We ship that inside a lead housing we call a generator. And if you ship the radium, you can then have daily purification of the lead isotope. This is in contrast to a lot of other isotopes. So if someone that's going for a PET scan, they use something called fluorine-18. That's made in a cyclotron every day. A cyclotron spins electrons at 10% of the speed of light, smashes them into each other, and you can kind of turn one element into another. That's a really cool way to make some isotopes. You can make isotopes in reactors. You can make them in other sort of devices too. But when you're thinking about how you kind of create your isotopes, you often create a lot of stuff that you may not need, and you don't want any of that inadvertently going into a patient. And so you really have to make sure that you're doing ultra pure isotopes into patients. So what's really cool, what we do is we buy ultra pure thorium-228 from the U.S. Department of Energy. It used to be a waste product that they had, and now it's actually being made purpose-built for our industry, which is great. They have a lot of material in hand. They can produce, for all intents and purposes, almost infinite amounts of it. And when they do produce the thorium-228, we access that, and then we have a controlled flow into down into the isotopes we're looking for. And what's cool about isotopes, that each one is chemically different, right? They're literally different elements. And so they're going to behave different chemically, which means we can, using very straightforward principles, sometimes it's charge, sometimes it's lipophilicity, separate out isotopes from each other using chromatography. It's a really interesting way to actually go using different kind of liquid affinities purify based on charge and things like that. So very elegant way to separate out different elements from each other and then only give a pure compound to your patient.
Got it. And speaking of elegant, we've talked about the radioisotope, and we'll put that over there for now. But there's a lot more here in terms of the modification to ultimately create a final product here that you've done, and that involves three other things: the cell targeting peptide, which is specifically involved in targeting the cancer. But then there's also this component, a chelator and a linker. The chelator, which correct me if I'm wrong, is about it binds the radioactive stuff and does some pharmacokinetic magic to it, so it doesn't go in the wrong places, and then the linker ultimately hooks that up with the peptide. Very elegant, but again, walk us through ultimately what the core product becomes with these four components to it.
Yeah, so when we actually think about, the smallest radiopharmaceutical is just an element, radioactive iodine. And so that's one that you can actually give, and it treated thyroid cancer starting in the 1940s with Saul Hertz. And he was able to actually give some really good therapeutics that way. The challenge is when you're actually trying to have the element go somewhere else in the body, you're going to try and turn it into a drug, either a small molecule or a peptide or an antibody. And each of those will then have impacts on biodistribution. And so ultimately, it's the biodistribution that matters. What does your body do with any substance you give it? If you give things that are much too large, right, they tend to accumulate in the liver and get processed that way. If they tend to have if they're sort of a charged protein, then the kidneys and the urine will grab those and process them. And so depending on how you change the overall design of the molecule, depending on what's there, the ultimate charge, the ultimate sort of what's inside each one, its physical size, all these things can tune it. But if you think about, everyone's heard of antibodies, and antibodies go and stick to tumors. When you inject an antibody, it's going to take about a week to actually get all the antibodies to clear out of the bloodstream and accumulate onto the tumor. But in our case, we're fighting half-lives of radiation, so we don't want to wait a week. We want to go really, really quickly. So an antibody is a really large, clunky molecule, and we basically say, let's take just the sticky parts. And the sticky parts are little peptide fragments. And we figured, let's just take some of those peptide fragments. So only use the sticky part of the antibody and use that then to become the drug. And so if we combine the peptide fragment with the radioactive metal, but hold the metal in a cage called a chelator, then we can drag the metal against its will wherever we want to go. In our case, we want to go onto the tumor and not to go to other parts of the body. Along the way, what we found is that if you use a generic chelator, it's going to have a charge on it, and a charged protein fragment will get picked up by kidneys. So by making our compounds net neutral means the kidneys don't grab onto it and hold it, and therefore we decrease the amount of time that it may potentially damage the kidneys.
Got it. Perfect. All right. So now with all of that, let's talk about some of these targets, because one of the important baskets in your portfolio are these neuroendocrine tumors, and specifically, we've not talked much about these, but there are these heterogenic malignancies that originate from both neuroendocrine cells. So something like 12,000 new cases each year, 170,000 people living with them currently. Walk us, maybe just introduce us to neuroendocrine tumors, and then a little bit of sort of the subset of that, which I know you're focused on, somatostatin receptor subtype 2, which does some stuff to the receptor that makes it a little more easy for you guys to see it and target specifically that part of the tumor. And I'll shut up now and let you run with that one.
Sure. So, it's as I explained to my kids the other day, I said, there isn't a single, tissue in the body that can't get cancer. It's pretty wild. Like anything can get cancer, depending on where it starts. And it's really important for the doctors to know where did the cancer start. Because if you know where it started, then there's indications as to what's more or less likely to work, what's more or less likely to stick to it, how quickly will it grow, what does it mean for the patient's overall potential to survive. And so it's really important that we can get it right. With the neuroendocrine tumors, these are tumors that start in various glandular organs, so things that produce hormones, and not just the sex hormones, but a lot of the other signaling hormones in the body, like cortisol and things like that. And so depending on where they start, if they start to go out of control, then that's where it gets tricky for the patient. And so a lot of times patients with a neuroendocrine tumor they'll feel really gross out of the gate. And that's a technical term, obviously, feeling really gross. But what it means is that your hormones are out of whack. You're going to feel flushed and nauseous regularly, and there's a lot of symptoms that show up. And so the first thing you'll do, excuse me, with these, the patients start to feel really gross and really bad when they have a hyperactive tumor that's secreting hormones. And so what we're able to do is actually treat those patients on their symptoms only using a somatostatin targeting agent. The neuroendocrine tumors explicitly upregulate the somatostatin type receptor. And somatostatin type receptor has a lot of different epitopes, numbered 1 through 6, and there's all kinds of sub things. But the somatostatin receptor type 2, so SSTR2, which is what we call it, is heavily dominant in neuroendocrine tumors and a lot of other sort of rare tumors like pheochromocytoma, paraganglioma, neuroblastoma. And so a lot of interesting different tumor types that can show up, but they all happen to express SSTR2. And in our case, we have a lot of efficacy with what are called GEP nets, so gastroenteric pancreatic nets. And so these are neuroendocrine tumors that start in either the gastrointestinal system, in the bowel, for example, or they'll start in the pancreas. And so these are not necessarily colorectal cancers. These are secretory tumors that go out of control. And so it's, there was a classic case of this where Steve Jobs had a pancreatic neuroendocrine tumor. He didn't have pancreatic cancer the way it's normally described. He had a pancreatic neuroendocrine tumor. And so these tumors, you usually with patients, you'll try and keep them, stable on the, usually they come from the gut. They can show up in the lung. They can come from the breast. They can come in angiomas, the head and neck. So a lot of different tumor types are categorized as neuroendocrine, meaning they're highly secretory. And that if that's the case, they usually express SSTR2. And so the cool thing is that there's approved drugs that are imaging drugs that image if the patient has this SSTR2 overexpression. And what's so important there is that we can actually figure out if the patient is likely to respond to the medicine. Because if you have a medicine that targets the receptor, and without cutting the patient open, we can image if they have the receptor, you have a very elegant way to actually go and tell which patients have a higher probability of responding to the drug. So it feels almost obvious, like if they don't have the receptor, then they're not going to benefit from the drug. But the nice thing too is we don't need to do a tissue biopsy to actually figure out if they have the receptor. We can do an imaging scan.
And it's just continuing along that. So, I mean, I know you have some phase 1-2 work going on right now that's either enrolling or in process. Could you just say a bit about sort of where that all stands specifically as it pertains to these neuroendocrine tumors, and then we'll move on to some of the other programs. But we'll point in, obviously to the portfolio page in the show notes, but take us down, what's happening clinically here, because I know you got a lot going on.
So in cancer therapies, as a lot of your listeners may know, you don't jump into phase 1 in healthy patients, right? You're only doing that in patients with cancer because of the highly damaging potential of these medicines. And you often start with called dose finding. And so, usually it means you're escalating, but sometimes you'll de-escalate. So that's why you're dose finding. And you want to try and find out what's the right window. We're chasing something called a therapeutic window, which is, is there a certain minimum threshold where we can start to get the drug to work? And is there a maximum level that we shouldn't go over? Otherwise, we're going to cause more damage than we want. And so it's finding that therapeutic window. Not all drugs have one. Some are razor thin, like warfarin and some of the other blood coagulation things. Some are fairly broad, like aspirin or Tylenol. So we're trying to go for the therapeutic window. And so in this dose escalation study, dose finding study, we started patients at 2.5 millicuries. And because it was so safe, we jumped really quickly up to 5 millicuries. So twice as much. And at the 5 millicurie level, we've now treated 46 patients with that. And we then actually wanted to try a little higher. We liked the results we saw at 5, but we also wanted to test what happens at 6. And the FDA had previously looked at the radiopharmaceutical field and told us, we're not comfortable with you guys going up really quickly, because if you get into a level where things are dangerous, we need to be able to do that before a lot of patients get touched with any, and it's not just us, it's every radiopharmaceutical has this threshold. You want to go really, really carefully, because before you cause permanent damage. So we're actually showed all the fantastic, safety data to the FDA. We said we think it makes sense to go 20% higher, up to from 5 to 6. and they agreed with us, which is great. So we're exploring also patients at 6 millicuries. So we've got 2 patients dosed at 2.5. We've got 46 patients dosed at 5, and we're doing a few more patients at a little higher to see if there's any kind of difference. But the good news for how we communicate what we do is that that 5 millicurie dose had a really long duration of response. In fact, even those patients that were dosed at 2.5, they're almost 2 years out now, and we're still seeing stable disease there. And so someone with a neuroendocrine tumor, they're going to have a highly secretory tumor. They're going to feel really, really bad. And then they're going to be stable, managing the symptoms only, and then they'll start to progress. And then when they start to progress, that's when you seek a therapy like ours. And so if they progress, you want to bring them back to stable. And so that's the therapeutic endpoint. You're trying to get stable disease. It'd be great if you can actually get tumors to shrink, but you're targeting stable disease. And we've seen that the 2 patients at 2.5 millicuries now at kind of almost 100 weeks are still stable, which is fantastic. The patients at 46, a lot more patients there. We have patients that have what I call a durable response. We're tracking them through over time. We don't have 2 years follow-up because we only started the study a year and a half ago, right? And then the last patients came in 6 months ago, so we have to kind of track those patients through. But what we showed 2 weeks ago at the ESMO, the European Society of Medical Oncology conference, was that patients that were treated with our medicine appeared to have a very, very good safety profile. When I say good, that's always relative, but meaning that we didn't see any major adverse events. But really importantly, we didn't see things that were dose limiting. And so we had no dose limiting toxicities. We didn't have any discontinuations of the drug. We didn't have any grade 4, grade 5 adverse events. We didn't have any myelosuppression. And so we had a very, very manageable safety profile that we think really is able to give an interesting therapeutic alternative for patients. And we're being rewarded with this with a lot of physicians and patients asking for access to our medications and to our studies. We can only give these medicines within the context of a clinical trial. This is still investigational. And physicians that treat these patients have their choice of clinical trials. And the fact that they are really interested in our therapy being a potential alternative is really rewarding for us.
It's interesting because you also have the melanoma work going on, and the interesting thing about that, not just because it's a really tough cancer, but you have both monotherapy work going on, and you have the combination with the checkpoint inhibitor, and it's quite interesting from the perspective. We've heard previously about in this era of living drugs and CAR T and all that, especially and then obviously these drugs that affect Tregs and all the aspects of the immune system. And you pointed out early on the importance of the immune system in all this. We got to be really careful ultimately when we're bringing together now immunotherapy and radiotherapy that we're not causing harm to sort of the underlying sort of microenvironment and all that. That's very important in sort of the pathology here. And I'd love to if you could talk a little about the melanoma program, and again any interesting things with regard to combining immunotherapy and radiotherapy in this nature.
So it's an interesting question and an interesting angle of research that we're exploring. We received quite a few grants from the National Cancer Institute to try and research this a bit further. What we find in melanoma, we did some control studies, and we have a slide on our website that describes this mouse data. But we found that you always go against mice with melanoma, and you're going to say do nothing and see what happens, and they inevitably die. And so, if you're going to do an intervention, can you sort of stop them from dying? Can you sort of let them live longer? What does it mean if you let them live longer? All things so that you try and learn more before you go into humans. And what we found is that there's a certain kind of medication you can give a patient called a BRAF inhibitor. And if you give mice with melanoma a BRAF inhibitor, you actually end up extending their survival a little bit, but they inevitably die. But when we combined our drug with a BRAF inhibitor, we had a huge extension on the mouse life expectancy. And so the combination was super potent. And we then said, all right, well, let's do some more work. And we tried doing that same kind of research. We had mice. We saw that they had melanoma tumors that had metastasized. We tried giving ipi-nivo, so a dual checkpoint inhibitor, which is a regular kind of weapon in chemotherapy or in cancer therapy these days.
Yeah.
And if the mice are immunocompetent, sometimes they get resistant to checkpoint inhibitors. So then we tried giving just our drug on its own, and our drug did better than a checkpoint inhibitor. But when we combine the two, we got some pretty transformative results. And so combining our drug with a checkpoint inhibitor, it's almost like turning a cold tumor hot. And so what we end up doing is actually then having, the 50% of the mice survived and lived to the end of their lives without having the tumors. And what's more important, 3/4 of those, if we tried re-injecting new tumor cells into those mice, they could never regrow. And that was pretty amazing that you can actually teach the immune system to know what melanoma looks like, and so they can never get melanoma ever again. So pretty amazing ability, right? Not just are you treating the cancer, but you're teaching the body's immune system how to permanently fight off that specific cancer. And so absolutely fantastic data that triggered another grant from the NCI and to explore this further. And one of the reasons why we're so excited about what we can do here is not just can we cause some damage to cancer cells and less damage to healthy tissue, but we can actually train the body's immune system to permanently get rid of the ability to ever get that tumor ever again.
That's fascinating. Fascinating. Fits so nicely into this whole, era of, cancer vaccines and neoantigens and things of that. So yeah, it's a great finding, and it's nice the NCI is putting more resources into you guys for that alone. But there's one other thing is quite interesting. I wonder if you could take us down the path of your pre-targeting platform, because everything we've been discussing so far is what your perspectives, radio antibody drug conjugates, and it seems like you also have a process here separately looking at other types of solid tumors to sort of separate the components and use them sort of in a two-phased process to initially, I guess, target certain tumors with the linker or whatever, and then bring in the radio isotope later on. Can you talk a little about what this is all about and why solid tumors of this type are, amenable to this approach?
Sure. So we have this case where we think through increased complexity. In some cases, just the atom or the element is the drug. Like lithium is an example, radioactive iodine is an example. And then if you go from there into small molecules, that's interesting. Going bigger from that into peptides, and going farther than that, you get into the radio antibody drug conjugates, which are really, really big molecules. And so we've tended to focus just on the peptide part, which is great because we can show blood clearance within 30 minutes and then show really compelling, it goes to the tumor, it doesn't go anywhere else. Antibodies are interesting because they're so specific to tumor cells, but it usually takes time to get them to clear from the bloodstream. And so if you're actually going to give an antibody that's radioactive, you usually have to wait a week to see it clear out of the blood pool and then only accumulate and concentrate then onto the tumor itself. Okay. So for short half-life isotopes, that doesn't work, right? If the half-life is 10 hours, you can't wait 7 days because it's all going to disappear and the radioactive payload won't go where you want it to go. So we in collaboration with some scientists at Stony Brook, we identified a technology where we can take an antibody where it's kind of known what happens with it, and we can throw, and I'm going to use a terrible analogy, we can throw little sort of Velcro hooks onto it. And so we put a few extra little kind of hooks onto the antibody. The antibodies are massive. And so a few kind of hooks throughout that are just sort of, kind of 8 or 9 little hooks that are kind of scattered. They're really tiny relative to the antibody. And then give the antibody. It doesn't change what the antibody does. And so the antibody will then accumulate over a week. It's still not radioactive yet. It's still just accumulating on the tumors. And then we, kind of a week later, you give a little kind of peptide chaser, for lack of a better term, right? So you're going to give a little bit of a peptide, and all it binds to is those hooks. And so it's not going to do anything except for bind to antibodies with hooks on them. So what you'll have then is you have a cancer cell that slowly had all these antibodies start to attach to it and spread out from the surface. When the peptide goes in, it says, all right, I'm just going to stick to cells covered in antibodies. And therefore, you know how to target the peptide. So you're not trying to get the peptide to target a cancer cell with kind of variable morphology and biology. You're having it just going for really clear grappling hooks that it can jump onto. And that's a great technology to take the known biodistribution and specificity of certain antibodies, but marry that with the half-life that comes with a really short-lived radiopharm.
It's again, it's another very, very elegant approach, and I really enjoy listening to the way that you think about the pharmacodynamics of these different opportunities, and again, targeting a wide range of important tumors that can cause a lot of problems. So it's exciting. Taste, what else is coming up? I mean, clearly, I know you're going to be, there's some radiopharmaceutical summits in Europe early next year. I know we got the investor conferences. What's happening as it's public facing as we get into the early days of 2026? And then any other initiatives that we should know about while we have you today that are public facing that you can tell us? Anything I missed, please.
Yeah, no, thanks, Ira. I mean, there's so many exciting things happening at the company right now. We've got 3 programs in clinic. So our VMT AlphaNet program for neuroendocrine tumors. We have another program that we call VMT01 that targets certain melanomas and only if they express a certain receptor subtype.
Yep.
And our third program in clinic targets FAP alpha, which is targets a scaffold protein that shows up on the stroma of cells. So like the physical infrastructure that tumors will grow to protect themselves in the body, and we can kind of target that defensive mechanism. Each of those programs is enrolling patients right now. We're treating them, so it's reasonable to expect by sort of middle of next year we'll have clinical updates. We'll have what's happening in humans when they receive the drug in a controlled fashion. We think there's going to be some, there's a major cancer meeting in the middle of the year called ASCO. Early in the year, there's some other more specialized tumor conferences. So across those conferences, we'd expect to give updates on our programs. We're also building out our physical infrastructure. And so we're actually building facilities that can actually develop and produce these just-in-time medications. We have a really active research and discovery team. They're coming up with new peptides. And so we're actually looking to do first-in-human images on several different compounds next year as well. So in 2026, we're going to be seeing a lot. Our scientists are busy. We've got 170 employees around the world. And we have a great crew that's of basic scientists that are actually developing these novel new peptides, showing first-in-human work. We've got our production teams that are building out production scalability and capacity outside of our existing 2 manufacturing sites. And we have a clinical operations team that's executing against 3 different active INDs that we're enrolling in. And if the images look great, if the new targets in humans, we expect to initiate new clinical trials as well.
Awesome. Well, I'll be looking forward to seeing what happens in 2026. It's exciting to see how this whole area of radiopharmaceuticals has evolved from what it used to be. And again, you guys taking the lead in this fashion is really impressive, but really, really impressive work. Again, for everybody out there that is going to be listening to this episode of the show across the various podcast networks, or who will be watching on the YouTube channel, again, you've been spending time with Thijs Spoor, Chief Executive Officer, Perspective Therapeutics, doing really amazing work to develop these innovative radiopharmaceuticals for all sorts of cancers. Again, Thijs, I want to thank you for taking the time out to come talk to us and walk us through your story and everything you're up to. Thank you for everything you do for patients. And again, as we like to say on our show here, thanks for helping to create a better tomorrow for so many people via what you're doing. Really an awesome story, and I look forward to continuing to follow it.
Great. Thank you. I really appreciate you having me on.
Great having you.